Today I would like to discuss about the biological mechanisms that guide cortical patterning. As you know, the cerebral cortex is divided into different functional areas, and these functional areas are characterized by their input and output connectivity, by the layer composition, the layer organization, and by the composition of cells. Since the early depictions of different functional areas in the cortex that were made by Broca already more than 1 century ago, there is a lot of work that has done neurodevelopment to understand how a very thin and uniform neuroepithelium develops into an expanded cerebral cortex with very distinct functional modalities. So what we know about cortical patterning, or the patterning of cortical areas, also called cortical arealization, we know that the patterning of the cerebral cortex is determined by intrinsic mechanisms like genetic or molecular factors that already in the cortical primordium start with expression of morphogens from the cortical pattern, from the patterning centers such as FGF8, WINS, PMPs, and SHH. These morphogens, what they do, when they are released, what they do is to promote or repress the expression of what we call the genetic patterning genes. And here you have 4 of them: MEX2, PAX6, SP8, and CUPTF1, which is the focus of our work today, that is also called NH2F1. So this regarding the protomap hypothesis that states that genetic determination, intrinsic determination of the cerebral cortex is important for cortical area formation. In addition, we know, I should do this, right? Yeah. In addition, we know that extrinsic activity to the cerebral cortex, such as the input from afferent thalamocortical axons, also shape and organize the structural organization of the cerebral cortex. And for this, the visual system has been very useful in understanding how sensory input shapes the area formation in the cortex, and also when sensory input arrives, but also before the arrival of sensory input. The spontaneous activity before the arrival of sensory input is very important for the predetermination of retinotopic maps. So what is less known about cortical area, is it working? Sorry. I can't advance in my presentation. Hello?
Yes, we're here.
Yeah, sorry. Now it's working. Sorry for that.
Yeah.
Yeah, can you see my presentation again?
Little more.
No, you can't?
Sorry about that. I was away. Is there anything I can do?
No, it's fine. I think.
Now it's back.
Okay, great.
Okay, perfect. Yeah. So what we know about intrinsic determination of cortical areas? We know about extrinsic activity in coming into the cerebral cortex from thalamic input for cortical area formation. What is less known is what is the contribution of intrinsically generated cortical activity during development. From many groups, but here I'm just showing you the work of the COSAR lab. We know that in the cerebral cortex, early in development, there are events of calcium activity starting as sparse events as early as E20, that after evolve into organized patterns of synchronous activity, such as the ones happening at P0, the red events that are synchronous plateau assemblies with long dynamics. And this evolves upon glutamate synaptic connectivity into cortical early network oscillations here in green, that after evolve into GDPs, giant depolarization potentials, upon the development of GABAergic hub neurons. So what we wanted to address in this project is to see whether cortical patterning factors and the cortical patterning of the cerebral cortex had any link to this intrinsically generated cortical activity in the neocortex. So how do we address whether cortical patterning has anything to do with the patterns of intrinsic cortical activity in the neocortex? For this, what we did was to delete exclusively in the telencephalon with the help of the EMX1 CRE mouse. We deleted one cortical patterning gene that is typical for development, which is CUPTF1, also known NR2F1. In the absence of NR2F1, only in the telencephalon, again intrinsic to the neocortex, what we get is an expansion of the motor region at the molecular and anatomical level. We assess that the molecular markers of motor cortex are all over the place where the somatosensory cortex should be. And this expansion of the motor cortex in NR2F1 knockout mice occurs at the expense of the somatosensory cortex that then is ectopically then forming here in a caudal region next to the visual and auditory cortex. So what we get here upon the loss of NR2F1 is a totally aberrant patterning of the cortex. So to understand whether early intrinsic cortical activity is altered upon cortical patterning malformations, what we did was to address cortical activity events between P0 and P7 in the somatosensory cortex of controls, in the primary motor region of controls, in comparison with the region of the motor cortex that should be somatosensory but is then now motor, which we call, for simplicity, motorized somatosensory cortex, or MS1. So what you will hear across this talk is the characterization of many different types of activity and excitability patterns and profiles between these three regions: motor, somatosensory controls, and the motorized somatosensory cortex in mutants. To start understanding whether intrinsic cortical activity is altered, what we did was to perform calcium imaging in horizontal slices from control and mutant mice at P1 and P4, using the calcium indicator BAFTA during 15 minutes after 1 hour incubation. You see here maximal projections of the BAFTA positive cells in these three regions, in motor, somatosensory in controls, and MS1 in mutants, and the calcium transients of cells that were selected manually. What we observed when we compared the postnatal day 1 to postnatal day 4 stages was an increase from across time of the frequency of calcium transients, so what is expected, and also an increase in the index of correlation between the calcium transients, which is indicative of the degree of synchronicity across neurons in the network, which is also expected as maturation of the cortex progresses. But specifically, when we compared the MS1, the motorized somatosensory cortex, that is N2F1 deficient, to the motor one and somatosensory cortices of controls, we observed a slight and very subtle increase, but significant increase in the frequency of calcium transients in the mutant cortex compared to the other control structures, as well at P1, with no difference in the correlation index, but at P4 we observed some differences between, first between control motor and control somatosensory cortex, but only between, the motorized somatosensory one when compared to the normal somatosensory cortex, indicating that some characteristics of the functional organization of the mutant cortex resemble the motor one, but on the other hand, some activity, hyperactivity was displayed early on at P1 in the motor mutant cortex when compared to controls. We were wondering in this, after these experiments whether any of these phenomena were maybe due to the adaptation of incoming stimuli. Again, what we have is a recombination of CUPTF1 only in the cortex, but still we have a setting in which incoming fibers from subcortical nuclei were present. So in order to understand whether intrinsic activity in the neocortex was altered independently of the incoming input from other structures that are still like normal, like where CUPTF1 is expressed, we thought it would be a valid approach to dissect the cortical regions of the different cortices and plate only the cortical neurons in microelectrode arrays composed on 59 electrodes at EF 18.5 and let these cultures mature until we see synchronized activity. So when we did this, we saw that in the motor 1 control cortex, the first time point at which we observed activity or coordinated activity was at day in vitro 9. And at this stage, we observed that the motor cortex was having a higher frequency of events, of synchronous events, when compared to the other two structures, either somatosensory control or motorized somatosensory mutant cortices. Still, even though the difference between motor and somatosensory were significant, still the cultures containing neurons that lack NR2F1 were the ones displaying the lower number of frequency in this setting. When we analyzed each type of event, each type of burst, how many neurons were contributing to this burst, we observed that in every burst, in mutants, the number of neurons contributing to the burst was significantly higher when compared to both control conditions, and also the cross-correlation index was significantly higher in the mutant scenario when compared to normal cultures, suggesting that probably in this intrinsic cortical activity was more synchronized in the absence of NR2F1. And the abnormal cortical activity, the first mechanism that we could think about that could contribute to this was the expression of ion channels and therefore the intrinsic excitability. So we hypothesized that if cortical activity in cortical neurons was altered, was functioning in an abnormal manner, a possible hypothesis would be that the intrinsic excitability of pyramidal neurons would be altered. And for this, when we decided to assess for intrinsic excitability of neurons, we had to choose for the same type of neuron across cortices. As you know, because it's necessary to compare intrinsic excitability or intrinsic properties between the same type of neurons. Therefore, we chose to assess intrinsic excitability from layer 5 pyramidal neurons, because layer 5 is present both in M1, as you know, as S1, and also in the MS1 of the mutant mice. You can see here the typical morphology of layer 5 pyramidal neurons, and in the lower part, in the lower panel, you can see the typical electrophysiological profile, regular spiking neurons, characteristic of these cells. In an overview, not much was changing, right? But when we analyzed carefully some properties such as the response to hyperpolarization, we observed that both motor as well as somatosensory neurons in control mice were displaying a SAC response after hyperpolarizing current steps, but mutant neurons that were deficient in NR2F and where in a cortex that is abnormally patterned, we're losing this SAC response. You can see the quantification here. And what we also found were differences in some intrinsic properties, like the minimal current that you need to elicit an action potential in mutant neurons from the NA2F1 mutants when compared to both control conditions, as well as resting membrane potential when compared to somatosensory cortex.
Is that, sorry, I think your screen is kind of frozen because it doesn't change slide anymore. We are still in the bursting activity in isolated slices.
You didn't see this?
It looks like it's frozen. We don't see when you change your slide. Maybe. Okay, so now that's good. Move.
I will share again.
Yeah.
Now?
Okay, so now we still see the bursting activity. Yeah.
Okay. Oh, so should I go back? Should I?
No, that's okay. This one is good. And so. Okay.
So here we have the quantification where we observe higher number of spikes in each burst and high correlation.
Yeah, this one, yeah.
Okay, so yeah, here you have again the morphology of layer 5 pyramidal neurons, the electrophysiological profile. I was talking about the SAC response in motor 1, primary motor cortex, somatosensory cortex, and the lack of SAC response that is quantified here on the right. And then we also observed some changes in specific parameters like the Rio base that is reduced in mutant neurons compared to controls, as well as resting memory potential that was clearly significant between these two, but not majorly changed compared to motor, suggesting that in these neurons, even though the major features such as the electrophysiological profile, input-output function, many other properties that I don't show because they didn't change, there were specific electrophysiological properties that did change. In addition to that, we also, obviously, we reconstructed the morphology, and we found that this, the morphology of the cells, the complexity of the dendrites was much reduced. It's something that we see at the level of Scholl intersections. You see that the profile is much reduced in mutant neurons when compared to both types of controls. And this was primarily due to a reduced number of primary dendrites, of basal branching points, and basal tips of these basal dendrites, suggesting that together with the changes in excitability, we will also see changes in the structure and the morphology of these neurons. Accompanying the changes in the structural morphology, when you think about structural changes, you could easily think about structural configurations that change in response to changes in excitability. So it was quite logical to, after think about possible changes that could occur at the level of the action in initial segment, since, as you know, that a change in the excitability of a neuron is changing the structure and the position of the action initial segment. So we use these markers for action initial segment and killing G in layer 5 neurons that are labeled with CTIP2. And we compare between somatosensory, motorized somatosensory neurons that are lacking NA2F1 in an abnormal patterned cortex, and the control somatosensory one. We observed a reduction in all measurements we did from the AIS, in the AIS length, diameter, and distance. And after this, we thought this could probably be due to a delayed maturation in the formation and in the development of neurons. And as one possible approach to check whether there is a delayed maturation in all of these properties would be to test whether these properties remain or not altered at later stages, like at P21. So when we check, for example, for action initial segment properties or structure at this older stage, we saw that the changes in AIS measurements remained, indicating that there was no delayed maturation, but the neurons remained altered across development. In addition, we assess for IAS plasticity in order to know whether there was plasticity still happening in mutant neurons. And for this, we applied the polarization protocol in brain slices at P7, after which we observed that in control neurons, the increase in excitability leads to AIS reduction in the length of the action initial segment. But in mutant neurons, we didn't see any plasticity issue. If the action initial segment was already short, we couldn't shorten it more by increasing excitability with this protocol. So, what would be the molecular underpinnings of this abnormal cortical excitability in an abnormally patterned cortex? To answer this question, we sought to perform a qPCR from P7 cortices for 33 candidates, 33 channels, ion channels for calcium, for potassium, for sodium, and glutamate receptors. From the 33 channels, we observed a decrease, a reduction in the expression levels of 7 channels. You have here depicted, and the most pronounced change in expression was observed for the HCN1. Interestingly, this HCN1, it's a hyperpolarized cation channel, is one of the major contributors to the SAG response that I was showing just short ago, in a couple of slides back. So we got very interested in this candidate, and we wanted to validate whether the expression was indeed reduced by performing immunohistochemistry for HCN1 in layer 5 neurons that are stained with CITP2. You can see here the colocalization and the levels of expression of HCN1 in a control condition. The levels of expression of HCN1 were indeed reduced. I don't have the quantification here, but it's in the paper. And this we could also validate through Western blot, in which we just loaded all the cortical tissue. So indicating that the reduction in HCN was not specific for layer 5 neurons, but was a generalized phenomenon probably happening in a majority of neurons. If we want to see this difference, it's probably happening in many neurons. We didn't check for the rest of layers, the cortices, but it's a strong effect, as you can see in the quantification.
Sorry, Isa, 3 minutes?
Yeah. 3 minutes? Good. So after observing these changes in ion channels, specifically in HCN1, after deletion and loss of NR2F1, our next question was to address whether NR2F1 was having a direct effect, a direct role on the transcriptional control of these ion channels. And for this, we looked for conserved motifs that could possibly be binding sites in the promoter for NRF2. We use this ECR evolutionary conserved browser to identify this this domain that is characteristic for the binding of NR2F. And for this, you have also these two bioinformatic tools. Specifically, this Chrome analyzer was developed in the context of understanding, in this paper, the binding sequences binding to NR2F1. And yeah, we identified this conserved motif and very similar motifs, but not the same, in 2 additional channels. So in order to validate whether NR2F1 was indeed regulating directly the expression of HCN1 and other factors, what we did was a chromatin immune precipitation, which resulted in a positive pooling of the HCN1 promoter and indicating that indeed NR2F1 is regulating directly HCN1, but not other possible ion channel candidates. So I guess I'm about to finish. I show you that cortical transcription factors that regulate cortical patterning are influencing intrinsic cortical activity through the expression of voltage-gated ion channels that determine the intrinsic properties of neurons. And with this, what we did is to provide a pioneer link between cortical patterning specification and the emergence of early development, intrinsic cortical activity during development. This is relevant for the future objective of my lab, together with the collaboration of the Michel Stude lab and Xavier Lain-Cuquel, because we are trying to understand better the implications of NR2F1 for neurodevelopmental disorders. And specifically, this predisposition on this specific gene is leading to a very rare syndrome that has the name of the medical doctors that put this name, which is Borg-Hoornstra-Schaaf optic atrophy syndrome, with a cluster of symptoms that are from epilepsy, autism, to obviously the visual impairment. What else? What is the future of our future goal with preliminary data? So we are focused on the neural circuit basis of neurodevelopmental disorders, specifically autism, for example. And we always have been, trying to assess whether, cortical, striatal coupling is responsible of repetitive behaviors, which some of them you can see here in this NR2F1. But in this case, I think our question it's very exciting for us, because what we have here is a totally abnormally patterned cortex. And what we find very interesting is to understand how many functions are conserved in these mice, and which are the autistic traits that we can reproduce using this model, bearing in mind that the cerebral cortex is totally screwed up. So you can see the exploratory, we started doing analysis of exploratory behavior of NR2F1 deficient mice, and which have a very particular phenotype, despite, in addition to this jumping phenotype on the corners. These mice have a clear hyperlocomotion across days and in every possible condition. So yeah, we are very excited to dig in the future more about these the subcircuits that are responsible of this hyperactivity. And I would like to finish by thanking all the people that did the work with me in France, and all the people that are now contributing in Spain to progress on this project, together with my collaborators on this front. Thank you very much, guys.
Thank you, Isabel. And now it's time for questions.